For the approximate century and a half since Charles Darwin discovered evolution and Gregor Mendel laid the basis for genetics, our picture of the evolutionary tree and the linearity of evolution as a whole has remained rather constant in the scientific mind. But what if there were a mechanism that could throw a wrench in the machine, could distort the view of inheritance? Well, that mechanism is lateral gene transfer, and it could completely revolutionize the way in which we think of evolution. But before we define it, we need to dig a little bit further into biology. One rather uniformly accepted idea in biology is the universality of the genetic code. No matter how differently evolution shapes us as organisms and species, we are all composed of the same amino acids, coded for by the same DNA. Whether you are a bacterium, or a bean sprout, or a baboon, your life is dictated by 4 letters: A, C, G, and T. This principle is the basis for genetics, and it links the most basic organisms to the most complex. As any biologist will tell you, there are 2 ways in which any organism may get its genes. From linear reproduction, whether it be binary fission, meiosis, or mitosis, or if you are a bacterium, from plasmid recombination. Okay, so for the most part, evolution is still good and simple. Offspring get their genes from parent generations, and plasmids are usually only spread between bacteria of the same colony. But wait, there's one more way. Enter lateral gene transfer. Lateral gene transfer is the movement of genes between organisms of different species, and it takes on 3 forms. First, there's prokaryote to prokaryote lateral gene transfer, which is carried out either through interspecies plasmids or by transduction through bacteriophages, which means that a bacteriophage takes up the host DNA instead of its own and infects another host with that same DNA. Then there's eukaryote to eukaryote gene transfer, which can be mediated by relationships like that of lichen or of the popular endosymbiotic theory. But then there's prokaryote to eukaryote gene transfer, which is most commonly a result of some sort of symbiotic relationship. And we've seen it in nature in such cases as Wolbachia pipientis in the Chinese bean beetle, but it has so far remained elusive in its mechanisms. And it really complicates evolution. But perhaps if we think outside the box, we can hypothesize its mechanics. One promising theory involves the humble bacteriophage, infector of bacteria. Now wait a second, you may be thinking to yourself. It's a bacteriophage, and aren't all viruses host-specific? To which any biologist would answer yes. But, and stick with me here, we all know that nature's in no way perfect, and so even if viruses are host-specific, is it too far-fetched to believe that a virus could mutate or make a mistake? Enter now the flagellotrophic bacteriophage. Breaking that down, we see flagella and tropic, which we can understand as to be attracted to flagella, which are whip-like appendages used by microorganisms to move around. This means that there's a phage that will infect bacteria based on their flagella rather than an indicator on the cell membrane. Now we have room for biological error. In even the simplest of multicelled eukaryotic organisms, take the nematode for example, reproduction involves a very important cell by the name of spermatozoa. And what is one characteristic about spermatozoa that is unique to it? Well, it's flagella, of course. So now, imagine you have some sort of complex eukaryote, for example, a male deer. And inside that deer's gut there are bacteria which aid in the process of digestion, a very clear symbiotic relationship. At some point, the bacteria are infected with a flagellotropic virus, but rather than creating new phages with phage DNA, they accidentally extract a gene from the host genome, say, for digesting large sugar molecules. Now let's say a few of those new bacteriophages end up in the reproductive organs of the deer. Here the bacteriophages find spermatozoa which, to them, may appear to be indistinguishable from their normal host bacteria. The cells are then infected, and the gene has a chance of being stored in the cell genome. So what you could end up with now, if the deer were to successfully mate with a female, and the spermatozoa were to fertilize an egg, is an offspring of the deer which has a gene for digesting large sugar molecules, which no deer in the species previously had. And this sort of transfer could be applied to any case in which you have some sort of symbiotic relationship between a bacterium and a regularly reproducing eukaryote. The natural opportunities suddenly become endless. So what then are the implications for us? Well, it means we have to rethink evolution. We have to redraw the evolutionary tree. Instead of having one tree which has very separate and distinct branches, it may look a little bit more like this. Confusing, huh? It's almost as if we're grafting branches on the tree of evolution. And as much as it is confusing, it's equally fascinating, because now, with the discovery of lateral gene transfer, we can form a much better and more intricate picture of the universality of nature and the relationships that it allows to be formed between species. And when you think about it, it almost reverses evolution, and that species are no longer growing farther apart, but rather closer on our now much more complicated evolutionary tree.